Class, can you hear me? Hi, very good evening. Could you confirm me that if you could hear me properly and join it via the link? I cannot send messages in the due to some error. Slow mode is enabled. You cannot send messages. So what is slow mode? Admin, please remove the slow mode for me. Can you hear me, everyone? So I'm trying microphone today so that we can have less noise. Please, where is ma'am? So that's the thing. Ramneet ma'am gonna take the classes of Khalsa College. Ramneet ma'am gonna take the classes for the College especially, and I will take this class. So please ask for the ma'am's link then. Okay, let me talk to the admin. We start as soon as you all are joined us. So it's 77, at least 100 students. हाँ जी सर, टेलीग्राम पे दिया सर. सॉरी, कौन से में सर? मैंने सर टेलीग्राम में शेयर करा है. हाँ, बच्चों ने मेरे को सर स्लो मोड में रखा हुआ है, आपने मुझे स्लो मोड से हटाइए प्लीज़. कोई स्लो मोड होता है सर, वो एडमिन हटा सकता है. हम्म. जी जी सर, लिंक तो शेयर. तो उसमें व्हाट्सएप वाले ग्रुप में भी शेयर करना है सर? ओके ओके. So shall we start, students, or we wait for further just 5 minutes more, then we start? Okay. So we did last time till DNA fingerprinting. So let's go with the continuing with the recombinant technology and continue with its friction enzymes. So how their naming is done, what are the various types of friction enzymes. So it will be subjective also. So these three subjective has to be read along slides. So these three chapters, DNA fingerprinting and DNA based markers. Once we are done with them, today we will deal with the protein isolation, purification and quantitation. So how to isolate, how to quantify it, then how to purify it. So all these tasks will be done in the next case. So that will be more interactive with a lot of videos and animations. So please, before we start, that's how things we're gonna do. So there are two groups were created, one for the Khalsa college and one for the other students, because they so whosoever wants to join me here, they can join me here with this part. Others can switch to the other ma'am, Ramneet ma'am, and can join it the same course. So I will continue with my same teachings as I was about to teach you today. So today is day 5. So I will start then. I hope there is no doubt anymore. Everything is clear. So mainly restriction enzymes, as we know, they help in the restriction digestion of your enzymes and restrict them from the particular sites, creating sticky and blunt ends, and then which could help in further cloning. And this lecture has been dedicated to them only, to restriction enzymes, because I felt while we teaching you all the cloning techniques, molecular biology techniques, um, so these restriction, something about a little bit about restriction enzyme is also required to teach you about. So that's why I kept this. So this is the part how the various appendix of this chapter, that is introduction, we will deal with mode of action, star activity, what is star activity, and we deal with the nomenclature, then various types of restriction enzymes, DNA ligase, kinase, reverse transcriptase, and clinophragments. So we try to cover all these parts. So to manipulate DNA into in vitro, uh, the ability of manipulate DNA in vitro depends entirely on the availability of the purified enzymes that can cleave, modify, and join the DNA molecule in the specific way. So we have the enzymes which carry out these functions of manipulating the DNA, and each enzyme has vital role in playing the process of genetic engineering. So various enzymes that are used in genetic engineering are nucleases, restriction enzymes, DNA ligase, kinase, phosphatases, reverse transcriptase, terminal deoxide transferase, or RNAPs. Nucleases are a group of enzymes which cleave or cut the genetic material. DNAs and RNAs are further classified into two parts, that is nucleases and DNases. So nucleases which cut or act on the DNA, that are classified as DNases, which act on the RNAs, they are known as RNases. So DNAs are further classified into two parts positions where they act. They could be N-terminal. If they are cutting at the N-terminal, they are called exonucleases. The one cutting at the non-specific regions, they are known as the DNA, called as endonucleases. So exonucleases requires a DNA strand with at least two 5' and 3' end, and they cannot act on the DNA which is circular. And endonucleases can act on circular DNA and do not require any free DNA ends. So exonucleases release nucleotides, whereas endonuclease releases your short fragments of DNA. Whereas restriction enzymes which act on the specific positions or sequence on the DNA are called restriction endonucleases. The sequences which are recognized by restriction endonucleases or restriction enzymes are called recognition sequences or recognition sites, and they are also known as palindromic sequences like TAAT, GATTA. So like which are repeating from the left side and the right side, they are the same. And further, different restriction enzymes present in the different bacteria can recognize different different restriction or same restriction sites. So only two restriction enzymes from a single bacterium will cut at the same restriction sites. So not from all the sites then. So their mode of action is mainly done with the help of recognition sites they find. If the methylation is done in the recognition sequence, then they just for the DNA and does not cut it apart from there. Then comes the star activity. In the star activity, it's mainly sometimes restriction enzymes recognize and cleave the DNA strand at the recognition sites with asymmetrical palindromic sequence. For example, BAM H1 cuts at the sequence GA and then TCC. But under extreme conditions, such low ionic strength, it will cleave only the following sequence like NGA TCCC, or like this. Such activity in the restriction enzyme is called as star activity. How do you name these restriction enzymes? How the naming is done? It's mainly done was given by the Smith and Nathans, and every restriction enzymes would have specific name which could identify it uniquely. The first three letters identify the biological source. That is, first letter has to be genus, and second letter has and second and third being the first two letter of the specific species name. So like Escherichia coli are called ECO. So first letter and the second two letter from the species. First from the genus, second from the species. Same from Haemophilus influenza then becomes HEN, and Diplococcus pneumonia becomes DPN, and so on. Then comes the strain of bacteria, which kind of strain it is. Then we give them by the name like R. If the strain R is, then E. coli R. Then comes the another part that which Roman numeral, like which strain it is. It is strain 1 or strain 2. Is it from the enzyme 1 or enzyme 2? Then we give them E. coli R1, E. coli R2 for the second. So like this we have these namings of the restriction enzymes. So how many types of restriction enzymes we have? The restriction endonucleases can be divided into three groups, that is type 1, 2 and 3. Type 1 and 3 have an ATP dependent restriction activity and modification activity, whereas the type 1 enzyme and type 2, type 1 is cutting at the random sites, whereas type 3 cleaving at the specific sites. On the other hand, type 2 is possessing a process separate enzymes for endonucleases and methyl activity, and the most widely used for genetic manipulation. So we will not go in detail. So these are so whatever we have studied so far, this is the right way of types of restriction enzyme and their differences. So in the structural part, enzyme type 1 has a complex of 500 to 600 kilodalton composed of three separate subunits, whereas type 2 is homodimer of 20 to 70 kilodalton, where type 3 is a heterodimer with subunits of 70 to 100 kilodalton. And the composition is multi-enzyme complexes with R, M and S subunits. So like R is for the endonuclease, M for methylase, S for specificity. And then type 2 is separate enzymes, endonuclease is a homodimer, methylase as a monomer. Whereas in type 3, M subunit provides specificity on its own, function as a methylase, heterodimer with R subunit function as methylase. And here the cofactors are magnesium, ATP, adenylyl methionine, that is SAM, that is needed for the cleavage as a methylation. Then comes the magnesium, SAM for methylation only, then magnesium and ATM and SAM for further in the type 3. So go through them. So these are some are the 4 base cutters, 6 base cutters, 8 base cutters. So some are 4 base cutters includes like this, ALUL, HEA3, TAI1, MBUL1, DPN1, SAU3A1. So which recognize these respective sites. They cut it from these parts, giving you blunt ends here, giving you sticky ends here. And these are the 6 cutters and these are 8 base cutters. Some examples. So DNA ligase is the one which joins through DNA, joining two different segments. So cohesive ends generated by restriction enzymes would be anneal them together with the help of forming hydrogen bonds, and this ligation is done with the help of by the enzymes isolated from the E. coli and T4 bacteriophage. So T4 ligase requires ATP as a cofactor and E. coli ligase requires NADP as a cofactor in their functions. Kinases is the group of enzymes which adds a free pyrophosphate to wide variety of substrates like proteins, DNA and RNA, and it uses ATP as a cofactor and adds a phosphate by breaking the ATP into ADP and pyrophosphate. So it is widely used in molecular biology, genetic engineering to add radiolevel phosphates. So phosphates could be of 3 types: alkaline phosphatase, bacterial alkaline phosphatases and calf intestine alkaline phosphatases. So within alkaline phosphatases, they are the one which remove the phosphate from a variety of substrates like DNA, RNA and protein. They help to remove the phosphate from your proteins or DNA or RNA and act on the buffers which act on the basic buffers pH 8 or 9. They are called alkaline phosphatases and most commonly known as bacterial alkaline phosphatases BAP and calf intestinal phosphatases CIP. So alkaline phosphatases are metal enzymes and has zinc ions in them, and bacterial alkaline phosphatases BAP is a dimer containing 6 zinc ions, two which are essential for enzymatic activity, and BAP is very stable and is not inactivated by heat and detergent. Whereas CIP is also a dimer, it requires zinc plus and magnesium ions for actions. CIP is inactivated by heating at 700 degrees Celsius for 20 minutes in the presence of 10 millimeter EGTA. Further reverse transcriptase, this enzymes use RNA molecule as a template and synthesize a DNA out of it and complement it to your RNA molecule. And these enzymes are used to synthesize DNA from RNA, and further RNA dependent DNA polymerase is known as reverse transcriptase enzyme. Then comes the last and two last types of enzymes that we were discussing. Terminal deoxynucleotide transferase, that is a polymer which adds nucleotides at 3' end and does not require any complementary sequencing, does not copy and DNA sequences. Whereas RNase-P, it specifically cleaves at the 5' end of RNA, and it is a complex enzyme consisting of small proteins, and it has a 377 nucleotide RNA molecules, and it has been observed RNA molecule possess at least part of the enzymatic activity of the complex. Hence, that's why it's called as ribozymes. So, what are clinophragments? Sometimes in your GATE or NET exams, it's coming because nobody goes through them, these parts. So, E. coli DNA polymerase 1 consists of single polynucleotide chain. They consist of single sometimes polymerase 1, DNA polymerase 1 from the E. coli, single polypeptide chain, poly 1, which carry 3 enzymatic reactions that are performed by 3 functional groups, 3 functional distinct groups. So 2 fragments are obtained from DNA polymerase 1, which is treated with a trypsin in mild conditions, and the larger fragment is called as clinophragments. And these fragments are of 602 amino acids length, and its function is to add nucleotides at the 3' end and 3' exonuclease activity. So that's the main function of them. So that's all very short and crisp. I don't want to go with the subjective things longer with you students because it might be heart-breaking. So these chapters we will focus more upon the more practical based. So far we had a quite good learning. So where is the objective? So we did the basics of MBBT. We learned about cell culture technique, microscopes. We have done various types of molecular biology techniques. We did recombinant DNA technologies. All the parts related to them we have done with that. Now today is the day 5th. So where we will be dealing with the protein quantification, separation and purifications. So we will be dealing with mainly this part today. So let's start with this without taking much time. So student who have taken part of proteomics, they will also learn these parts there also. So it's a common part in both, because protein isolation, quantification and purification will be coming all in all together in 3 parts. So in this we deal with the various assays, homogenization, fractionation, centrifugation, quantitations, chromatography, electrophoresis. So each part we will go in quite details, and electrophoresis, I think I will be happy to take you one day lecture with that, to deal with the various types of electrophoresis. But today we will deal with various type of chromatography, actually how various type of chromatography and separation is done in the field of protein field, like that is proteomics. So this is somehow is a combination of both molecular biology and biochemistry. Both are belonging to this technique. Mainly these are the chromatography belong to biochemistry techniques, but it also comes to the proteomics because they are all conjoined together. So everyone is dependent on each other. So let's start with today's lecture. So here we will be dealing with the Nobel Prize winners on the chromatography and electrophoresis. That is our Archer John Porter Martin in 1952 for the partition chromatography. Then Professor Singh also on the partition chromatography. They got the Nobel Prize winner. And Telius from Uppsala University, they researched electrophoresis absorption analysis on the serum proteins. So in 48 they got it. So in this year it's mainly the CRISPR is hot. So the CRISPR ones, people who have discovered CRISPR, they got the Nobel Prize winner. Then comes the mass spectrometry, that is sedimentation. In this case, Fan, Tanaka, Swedberg, so they all got this respective Nobel Prize winner in their field of the mass spectrometry and sedimentation. Then comes organelle fractionation to remove your different organelle like mitochondria, nuclei on the basis of fractionation. Albert Claude, Christian Dewey, George E. Palade, they got the Nobel Prize winners. So these are the famous people who worked really hard in the field. Then comes the protein sequencing. Frederick Sanger from United Kingdom, both they work on the protein of insulin, and other person work in the nucleic acid respectively. So they got their own Nobel Prize winner in their own field. Then protein assay. So it's a biological means used to monitor the presence of target protein. For example, enzyme activity, specific level functional proteins such as proteins and all these as a binding. So here we can see that lactate is producing pyruvate in the presence of NAD plus, and enzyme lactate dehydrogenase. So in this protein assay, we are converting lactate into pyruvate and leading to NADH and H plus signs later. And further with EL5A precursor we have here. So in the presence of two different enzymes with the synthase and hydroxylase of deoxyhypopysanine, and spermidine is added into that. So you are getting EL5 alpha deoxyhypophysail, and in this case hypophysail. So that's the difference of different enzyme producing different functions of protein. And how to isolate your protein? You can isolate your protein by using a different centrifugation, or first you have to homogenize. Homogenization is very necessary in that doing so. First you or how you say, like you do make a mixture for your masala, garam masala, the same way your cardamom gets out of it, the same way cell get disrupt and all the organelles came out from the cell part. So that is homogenization. Then fractionation is the crude separation of homogenates into fraction. So you make a fractionation, like different fractions are made, and then you remove each part as per the need. So homogenization could be done with the help of Downs homogenizer, sonication, osmotic shock, French press. So all these things are possible in the case of homogenization. In the case of fractionation, it is done with the help of pH or heat, ammonium sulfate precipitation it could be done, differential centrifugation it could be done. So once you have a homogenate, you put it into the forms of a centrifugal tube like this, and you centrifuge for 500 G for 10 minutes. So you have supernatant and you have pellet. In the pellet, you will have nuclear fraction initially. But if you go it further for 10,000 G for 20 minutes, you will have a pellet which will have mitochondrial fractionations. Then further in the cytosol, then you make a centrifugation, then you have a microsomal fractionations. So like this, if you keep going further, you will have the lower part, the inner part of your cells in more detailed manner. So in this way, you can isolate your concerned part. Beyond this part, you can isolate your concerned part of your cell by doing centrifugations based on the layer method, by differential centrifugation and rate zonal centrifugation. In the differential centrifugation, you have more dense particles and you have less dense particles, and you want to differentiate between them. So you go for centrifugations. The particles which are more dense, they will go at the bottom, which are less dense, they will remain up. So you decant the liquid into containers, so you have your all less soluble particle on top, and the all pellet will be down in that case. So rate zonal centrifugation, sample is laid on top of the gradient in this case. So you have your sucrose gradient, you take a sucrose gradient and then you put your sample on top. So you go for centrifugation. So there will be two kinds of particles, one with the less mass, one with the one with the less density, one with the more density, and they will differentiate like this. And then after you stop the centrifuge, and then you can remove your samples from the bottom or from the top by removing fractionation with the increasing mass of particles. So initially you will have less mass particles, then a little bit more, then heavier at the end. So in this way you can remove according to the sedimentation coefficient of your particle. So what is sedimentation coefficient comes now? So this is equals to the 10 raised to power minus 13 seconds. We should not go through in details about this formula. So this is the major formula looks like, it's good for your knowledge, but just remember that one sedimentation coefficient is equal to the 10 raised to power minus 13 seconds. So these are the some values have been told, like at 3000 G, 10,000 G, 40,000 G, for 200,000 G, at 40,000 G you can have proteins, large protein complexes, membranes, plasma membranes, mitochondria, large organelles, etc. With respect to the speeds. So the more the time is, more the speed is, the more different kind of organelles you can remove from your cell structure. Then comes the sedimentation velocity technique. In this is again like what we did in the rate zonal centrifugation. So you have your centrifuge tube in which you have a density gradient solution. So you put a low density and high density solution together. Then layer of sample on the top. Then go for centrifugation with the rotor. Then according to the sediment coefficient, your all samples will be layered. So we made a hole at the bottom, and then you start moving your samples as per the needs. So these are some density sedimentation velocity techniques. So they are according to the sedimentation coefficient and versus the density. So these are the various molecules are being separated together. So like chloroplast, mitochondria, nuclei, DNA, RNA, soluble proteins are all being separated like this. Now we did the isolation. Now we have to see quantification, like how much sample was there in our protein, how much did we extracted our protein in isolated. So we use, this is very crude method, it's not mostly followed in the big labs, but if you are in the short run, if you want to do some experiments, so this is the best method, that is Bradford protein determination. So Lowry protein determinations. So in this method, technique is simple and faster than the Lowry method. So there is a Lowry method also, which could help you to protein determination from 10 microgram to 100,000 microgram, but is subject to less interference. In this Bradford it is less interference, Lowry has a lot of interference in that. In this case, Commercy Blue G250 is being used to appear most readily to organolyzer residues, and this specificity can be lead to variation in the response of this assay to different proteins. So here you can calculate from 5 to 25 microgram of proteins in this case.
In this video, you will learn how to perform protein quantitation assay. In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, Read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So that was defined way to calculate your proteins via Biorate method. I hope all is clear, students. Okay. So you're all with me. That's great, great. So that was something about Bradford quantification. Now comes the next part. You can also do the quantitation with the help of ultrafluorescence protein-based quantitation. The method is given in the biotechnique paper, in which you add your sample dye, then you heat for 10 minutes, then you cool the temperature, and you can read the fluorescence of a sample, and that can let you know the how much fluorescence is there, and that fluorescence could equate to your protein microgram per ml. This is your ultra sensitive fluorescence based method quantitation. So you can quantify your proteins like this. Then there is a method. Now we are done with the quantitation. Now comes the next part, that is quantification. Purification. This purification method could be done with the help of technique used to remove small molecules from the protein solutions or to change the buffer solutions of the protein solutions. So dialysis tubing or bag is made up of cellulose and has different cutoff range. So dialysis bag, here you could see, um, have large molecules and small molecules. So there are concentrated solutions and other solutions, big solutions. So at the start of dialysis, you so everything is mixed together, but at the dialysis equilibrium is achieved. So large molecules will remain inside and the other will remove outside. So there is also protein Proteo Plus TM kit contains proprietary buffer and precipitation protocols to remove your samples out of your things. So let's check how to do this. So that was simple and quite to the point. How you have your uh, system in equilibrium. Initially, continuous dialysis is done. Semi-permeable membrane is there. Then you put in the H2O. Then you give water from the top. Then things are moved downwards, and then you have complete system of removing it with the level regulations. And then your samples are mixed, and at the end, what you're looking for is being removed out, and you have large particles at the end. So that was something about simple way of purifications. Now comes the protein purification with the help of liquid chromatography, which refers to as mobile phase is liquid in that case, and stationary phase is also liquid, or it could be solid also. So between the chromatography, first type that we're going to discuss here is gel filtration chromatography, in which you are separating your protein by size, gel filtration chromatography. So here you have a protein sample initially, and this is a molecular exclusion gel is there, and then this gel after some time which gives you three different colors, and three different colors are of high these gray beads are the large beads. Then you have these are your samples with the red, green and yellow. Yellow are the bigger ones, green are the smaller ones, which and the red ones are the which get trap in these beads. So these are the and these big beads are the carbohydrate polymer beads, and these small molecules enter the aqueous spaces within the beads, so they get entered to the beads, and whereas the yellow beads they just flow with the direction, and you have at then yellow and green left which are moving out. So these molecules could be separated first, then yellow could be separated separately, then green could be separated, and the red could be separated. So based on their size, these molecules could be separated easily. So, so in this gel filtration chromatography, you layer sample on the column, then add buffer to wash through the columns, then you collect your fractionations. So this is how done in practicality this gel filtration chromatography. You have a Cephedex G75 gel and you have Cephedex G10 gel, and then you add your samples, and then this is your HB, your different proteins, blue dextran, void marker 2 megadalton. Saffrose has exclusion limit of 4000. Cefacel has around 100,000, and then Cephedex dextran has difference according to it. So let's see Cephedex G50, how this is done in practicality. Let's go with this.
We're starting with gel filtration medium. This is Cephedex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the beaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now we're going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom.
Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of two molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1,000,000. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these two molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation is proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. It's still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
So you can see it was quite with the cephadex G50 we can separate these two different molecules, one with the light, one without light, and nicely separated like what we were discussing in the animations in the PPTs is done like this. So all these things will be shared with you, so don't worry. Now comes the next chromatography technique, that is ion exchange chromatography. So we were dealing with the based on the gel based so far. Now comes the next one is the protein in isolation that has low ionic strength and is added to the column. The protein column interacts with the beads via electrostatic interaction. So they are beads, so which are negative in charge. So to those the positive charge will attach and the negative will go downwards. So this way you can separate your molecules. Positively charged will bind to the negatively charged beads, the negatively charged protein flows through it. So you can separate your proteins based on that. So there could be cationic and anionic exchange chromatography. In anionic, the matrix carries a net positive charge which interacts with the negatively charged protein strongly, whereas in cationic the matrix is negatively charged and interacts with the positively charged proteins. So that's how the main difference between cationic and anionic is. And in ion exchange chromatography there is a group called sulfonate, quaternary amine, dimethyl methyl, diethyl aminoethyl hydroxyapatite are being used as a your beads, which are strong cation, strong anion, weak anion, strong anion respectively. So let's start with another video with ion exchange chromatography.
Ion exchange chromatography is a chromatography process that allows the separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into anion exchange chromatography and cation exchange chromatography. In anion exchange chromatography, negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged, and the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge, and at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge, and both protein B and C carry a net positive charge. In addition, protein C is more positively charged, since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also, varying the pH of the buffer can be used to effect the separation, and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified concentrated form.
So I hope it's clear. If we're dealing with unknown samples containing different proteins and we want to separate them, then how can we select the gel pore size? So according to that, for that, uh, so exclusion limit. So we have to check as per our gel fractionation range. So how much our sample has to be separated. We select our gel electrophoresis. Our gels like this size. So exclusion limits are given to us, the exceeded limits. So below that the proteins are, so we can do it. Otherwise, if this method is not good, so it's always the best method is liquid chromatography, in which you can separate your all size of particles via chromatogram of your C18 columns in a very well manner. So further students. So here we take just a 5 minutes break. So we meet at adding to how do we know which pH buffer is used in ionics if the proteins are unknown? How do we know which pH buffer do we use? Good question. So in case of some students are asking us that if the pH is unknown, then how can we separate them? How do we maintain the that was the question. How do we if the proteins are unknown? In that case, um, this one. So we choose according to the if pH is unknown, then we choose then in that case we go for another affinity chromatography instead of that one. So there are always some better method to go for. If this method is not working for us, we can choose another method. So in that case we go for affinity chromatography. This method relies on the properties of enzymes or proteins that you want to isolate. In this case, antibodies must be available that have been raised against the enzyme. An enzyme must be known to bind to some substrate such as ligand, non-covalent, irreversible way. And it is the most specific chromatographic method because it relies on the specific binding of the target protein with its own specific ligand. So in this affinity chromatography, glucose binding protein attaches to glucose on beads. So on glucose, our beads are being attached like this, and the use of glucose resins to isolate the glucose binding protein concanavalin A. So addition of glucose, so glucose binding proteins are released on the addition of glucose. So we added glucose, so these proteins are separated at the end. Concanavalin proteins. So in this affinity chromatography, you have protein recognized antibodies. The red ones are protein recognizing antibodies, blue ones are the recognized protein not recognized by the antibodies. So you wash them, elute with them with a pH buffer, and then you have your red ones separated and the white ones separated. So the drawback of affinity chromatography is to ligand or antibody can often be so strong that very harsh conditions are needed to elute the enzyme, and this leads to denaturations. And thus there is often limit to protein concentration that can be applied to affinity column. So let's check how the principle of affinity chromatography looks like.
Depends upon the reversible adsorption of biomolecules. Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and dilution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
That was a 3D showing of you that how could you, with the different pH you can separate the any unknown proteins very easily with these sample application wash, and with equilibration, sample application and wash, and an elution method. So like this you can do it. So now comes the next part, HPLC, that is high pressure liquid chromatography. So high pressure liquid chromatography is the enhanced column methods for protein separations. Resin size are more refined in this case. And all the method that we have described so far, HPLC column equipment is the best one so far. So in this case we have separation of peaks is done, that is the resolution, which is always better than the standard chromatography columns. Reason for this is that HPLC columns are packed with the much smaller beads than those used in the standard columns. An intermediate versions between columns and HPLC is called FPLC. That is fast performing liquid chromatography, but we deal with mainly HPLC in this case. So the column is a large tube containing small particles which has stationary phase in it, and chromatographic bed is composed by the gel beads alone with their inside the column. The sample is introduced into the injector and then carried into the column by the following solvent. Once the column, the sample mixture are results into the adhering to the diffusion into the gel. And the results found on the chromatographic bed by the flow rate with a different zone of sample of components, and these components are referred to as bands. So here you can see different bands 1, 2, 3, 4, 5 from your different solutions. So if you have mixture of 2 or 3 solutions, so each solutions will be seen as a peaks with a different absorbance with the time of 10 minutes. So HPLC is actually answers to every question. To discover that what is going on, which proteins belong to which. In this you have the peak resemblance belongs to specific proteins, and you can say, and it is separated very nicely in this case. So this is a highly nowadays there are also nano LCs are being used with the mass spectrometry, which is highly in demand.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper.
Oops, sorry.
In a mixture, to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase nonpolar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, the component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
I hope many of your doubts will be cleared by now. So here, let's take a break. I want break before also, but now it's, let's say, 36. Let me take your questions first. Um, if we are dealing with unknown samples, separate them. Yeah, videos will be shared in the presentation itself. So I can share my presentation now itself with you students. Reaction enzymes and protein. This will take some time. How to go back? Yeah. Yeah. Okay. By isoelectric focusing, we can find the isoelectric pH. In which regards you are saying this? Mainly, but LCMS is the best method so far. Otherwise, you can go for SDS page. With the SDS page, you can separate two proteins on the gel, and then you can work for your own specific thing. You can also go for 2D gel that we will be discussing between two different proteins. You want to separate unknown proteins. In that cases, you can also do. But in the known cases, we are as far we know, we will use these methods. Okay. Just give me 5 minutes break, 5 to 10 minutes break. So we meet at 40, we meet at 45, 18:45. Then we continue with other lectures. Yeah, from here. So So fast performance liquid chromatography is just where the solvent velocity is also controlled by the pumps, and the constant flow rate of the solvent is done in that case, and the solvents are assessed through the tubing through the outside reservoir. So now in the separation, the most effective techniques to get them into different sieve is a gel electrophoresis. Basically, in this, you add your samples and they run from negative to positive ends, and you have mixture of macromolecules in it in the electrophoresis, and the smaller molecules will go downer, middle will be in the middle, and the larger will be at the top. So you can separate according to that. So in the gel electrophoresis, you have acrylamide, methyl bisacrylamide, and they are joined together to make a new polyacrylamide gel at the end. And gel electrophoresis in the SDS page coated protein, place mixture on the proteins on a gel, applied electric field, cross-linked with a polyacrylamide gel. Direction of the migration is done in this way, and then the larger, smaller, and the smaller middle they are separated like this. And so here you can see SDS staining. Uh, all the molecules are being separated in different lanes, one with a small less protein, a bit more, bit more, bit more, bit more, less, and bit less. So this is a different condition, but the proteins looks like this, your separated proteins, and these are molecular markers at the end. So some other gels for data keeping in later part. This is C14 labeled crude cell extract flowgram, and these are some other cell stainings in 1 hour, staining with the timing with 1 hour, and then one with the de-staining, and then with the de-staining time 0 hours. So bands are clearly visible in the staining tray in this case. So these are three different kind of gels that has been used, stainings has been used: PS gel coat, blue staining reagents, and stain formation. So here are the best results so far in this case. Excellent results, clear, crystal clear background. Now after the SDS page comes the isoelectric focusing gel electrophoresis. So you run this gel in the 2D form in the first in this state where you have different pH, and on the right hand side is the lower pH, on the left hand side higher pH. When you run them under the electric field, so your lower pH will be on the right hand side, your higher pH will be on the left side. Somewhere in the middle with the isoelectric focusing which doesn't have any much the zero neutral pH, they will be remain in the center, and that's the basic principle of isoelectric focusing gel. So this is SDS page, looks like this, but in the IFE it's more prominent. You can see much better E alpha. The unmodified and unmodified one, we can see direct difference, 1 charge per molecule. More is possible in that case. So let's see how 2D gel electrophoresis is done. One principle behind it. So you extract pro to cell samples. Like this. Mix it up, vortex for 15 minutes. The proteins are mixed now, then you centrifuged. 12,000 G for 4 degrees Celsius for 10 minutes. Then you add chloroform, shake for 15 seconds. Incubate at room temperature. Centrifuge. Now you divide your supernatant into two new parts. Isopropanol were added. Discard the supernatant carefully. Acetone, vortex. Repeat this wash. And that's it. So can we store gels after we are done? Yes, you can store in the fridge at 4 degrees Celsius, and or at 20 or less than 5 degrees Celsius, in the buffer solution, so that they don't lose their textile, that's power to remain there. So in the 2D dimensional gel, you have pH 4, you have pH 10, you have isoelectric focusing like this. Then you separate your samples like this, pH 4 and 10. Then you run them into the in this state on the gel SDS space. So this is how you separate your 2D gel from your isoelectric focusing gel. Then you run them on the straight forwards. So this is how your 2D gel works like. This is two-dimensional electrofocusing. So you have a high pH here, low pH here, and on the side SDS polyacrylamide gel is working on this side, and this is how your samples looks like. On the top is isoelectric focusing, on the bottom is the SDS page. So high resolution two-dimensional electrophoresis of proteins being shown here. So this would be, I think, our last video of today. This will be a long one.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the IPG strip. The second segment will be on how to equilibrate the IPG strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the IPG strip. And I'm going to demonstrate this process using Biorad's 2D Starter Kit. The 2D Starter Kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, chaps, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 IPG strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has 2 sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip on the negative side. You'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the IPG strip is facing down, you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before this step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode with the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first, remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Bio-Rad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, Place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. Do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, flamingo and cypro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kumasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kumasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kumasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
So quite in detail manner about how to do 2D gel electrophoresis. The professor has explained us from the Biorad each part in detailed manner about that. So highly remarkable. So these are the results of the protein purification, single SDS. So this is the homogenate, salt fractionation, ion exchange, molecular exclusion. This is affinity. So affinity seems to be quite specific as compared to other microscopic technique. Then comes the end part, that is your MALDI. So sorry students. Just a second. Are these PCTs? What is PCTs and other stuffs made of normal plastic or other stuff? Ooh, I don't know what you mean. Could you be more elaborative, please? If you have no further questions, I can start. So MALDI-TOF is done after the LCMS is done, 2D gel is done, you have your samples, you got your protein samples, then you put them into this MALDI-TOF samples where you have your protein sample is ionized on a matrix. This is a matrix, you put your protein and laser beam is added. Then this is ion source where flight of tube is done. Light ions arrive at the detector first, then they will be encoded, laser triggers a clock, then they trigger and beam light into the lasers, and then you can check how things are. So you get different beams after the MALDI-TOF is done. With 1 2H plus ions, you have mass to charge ratio at the x-axis, intensity on the y-axis, and you calculate like beta lactoglobin protein has been calculated like this, and insulin has been calculated like this. So you have two peaks of your proteins. So this is how your two proteins are separated in the MALDI-TOF. These are some peaks of the same. So last video for the same.
Worker Maldi-Toff-Toff process. Maldi-Toff-Toff analysis provides additional structural information for target molecules. As Maldi-Toff is a two-phase process. Fragmentation can occur by application of higher laser energies, known as LID, laser-induced dissociation, or a background gas can be introduced to promote ion collisions. This process is known as collision-induced dissociation, or CID. Samples are co-crystallized with a suitable matrix on a sample carrier. The samples are vaporized by the laser into a vacuum while being ionized at the same time. During this process, the sample molecules can become excited. The now charged and excited particles are accelerated through the application of a high voltage, and fragmentation can occur as they travel through the flight tube. In this process, precursor ions and their respective fragments travel together at the same speed. Based on the expected time of flight of the precursor ions, specific ion packages may be individually selected, and all other ions blocked from continuing through the flight tube. The selected ions are then re-accelerated, with the precursor and fragment ions traveling at different speeds towards the reflector detector. The ions travel via the reflector towards the detector, and thus a Toff-Toff spectrum is recorded. The detection of the selected precursor ion and its fragments creates a Toff-Toff spectrum. Whether fragmentation occurs through the use of higher laser energies or a collision gas, the structural details derived from the Maldi-Toff-Toff spectrum provide a deeper analytical insight of the targeted compound.
Very well explained about these steps with the Maldi-Toff-Toff, how with the laser, how the fragmentation, how they are into ionized, how they are being separated, and everything is done in nice manner. So this is how Maldi-Toff-Toff is done. So that's it for today. Thank you very much. I already shared with the files everything, so please have a look. So we are done with day 5 with protein quantification, separation and purifications. Tomorrow we will deal with the electrophoresis, separation of molecules by electrophoresis. So tomorrow is the day 6. So see you tomorrow then. Thank you very much. If you have any questions, please free ask or write me back there. So I hope we had a good learning from these videos and lectures together, and going good. So students, I got to go now. See you then tomorrow, same time, same place. So we will meet at the right time at 5 p.m. Today I was just somewhere else for something, and yeah. Thank you very much. Bye bye. Dankeschön. Tschüss. Auf Wiedersehen. Take care.